Optimization of root exudate composition for improved nutrient absorption. The optimization of root exudate composition for improved nutrient absorption has emerged as a significant area of research in plant physiology, soil science, and sustainable agriculture. Root exudates, consisting of a complex blend of organic and inorganic compounds secreted by plant roots, play a crucial role in nutrient acquisition, soil microbial interactions, and plant health. Understanding and manipulating these exudates can enhance nutrient uptake efficiency, reduce fertilizer dependency, and improve crop yields in various soil conditions. This documentary delves into the mechanisms, composition, and potential strategies for optimizing root exudates to improve nutrient absorption, drawing on extensive scientific evidence, case studies, and experimental data. The nature and composition of root exudates. Root exudates comprise a diverse mixture of low molecular weight compounds such as sugars, amino acids, organic acids, phenolics, and secondary metabolites, along with high molecular weight compounds like mucilage and proteins. These exudates influence soil nutrient dynamics, microbial communities, and root soil interactions. Sugars like glucose and fructose are primary exudates that attract beneficial microorganisms, while organic acids such as citrate and malate facilitate the solubilization of otherwise inaccessible nutrients like phosphate and iron. Research by Jones et al. 2004 revealed that maize roots exude citrate to increase phosphate availability in phosphate-deficient soils. In contrast, legumes release flavonoids to initiate symbiotic relationships with nitrogen-fixing bacteria like rhizobium. The specific composition of root exudates varies across species, developmental stages, and environmental conditions, indicating the potential for targeted optimization to enhance nutrient uptake. Mechanisms of nutrient acquisition mediated by root exudates. Root exudates mediate nutrient acquisition through multiple mechanisms, including nutrient solubilization, rhizosphere pH modification, microbial interactions, and mycorrhizal symbioses. Organic acids such as citrate and oxalate chelate metal cations, making nutrients like phosphate, iron, and zinc more accessible. For example, in calcareous soils with high pH and low phosphate availability, plants like lupin increase citrate exudation to dissolve calcium phosphate deposits. Moreover, root exudates modulate rhizosphere pH to optimize nutrient solubility. In iron-deficient conditions, Strategy 1 plants such as sunflower release protons to acidify the rhizosphere and convert ferric iron, Fe3+, into the more soluble ferrous form, Fe2+. A study by Romheld and Marshner, 1986, demonstrated that iron uptake increased by 60% in sunflower plants exposed to iron-deficient conditions due to increased proton exudation. Interactions with soil microorganisms further augment nutrient acquisition. Root exudates act as chemical signals that attract plant growth-promoting rhizobacteria, PGPR, and mycorrhizal fungi, which enhance nutrient mobilization. For instance, leguminous plants exude flavonoids that initiate nodulation by nitrogen-fixing rhizobium, increasing nitrogen availability. Similarly, arbuscular mycorrhizal fungi, AMF, rely on strigolactones released by roots to form symbiotic associations that improve phosphorus uptake. Factors influencing root exudate composition. The composition and quantity of root exudates are influenced by various factors, including plant species, soil conditions, nutrient availability, and environmental stressors. Genetic variation among plant species results in distinct exudation patterns. For instance, maize predominantly exudes sugars and amino acids, whereas lupin secretes high levels of citrate. Environmental conditions such as drought, salinity, and nutrient deficiency trigger adaptive changes in exudate profiles to optimize nutrient acquisition. A notable example is the increased secretion of malate and citrate in aluminum-tolerant cultivars of wheat when exposed to acidic soils. Delays et al., 1993, found that malate exudation facilitated aluminum detoxification and enhanced phosphate solubilization, improving root growth and nutrient uptake. Similarly, phosphorus deficiency induces increased exudation of organic acids across various species to mobilize phosphate from insoluble forms. Soil microbial communities also exert feedback effects on root exudation patterns. Studies by Badri et al. 2013 demonstrated that exudate composition changes in response to microbial colonization, with plants exuding specific metabolites to recruit beneficial microorganisms while deterring pathogens. Strategies for optimizing root exudates for improved nutrient absorption. Optimizing root exudate composition to enhance nutrient absorption can be achieved through various strategies, including plant breeding, genetic engineering, soil amendments, and microbial inoculation. Traditional breeding programs can select for genotypes with superior exudation traits, while genetic engineering enables the targeted manipulation of exudate pathways. For example, transgenic Arabidopsis plants engineered to overproduce citrate exhibited a 40% increase in phosphate uptake under low phosphate conditions, according to Lopez Buccio et al., 2000. Soil amendments, such as the application of biochar or compost, can alter root exudation patterns by modifying soil pH, nutrient availability, and microbial activity. A study by Jones et al. In 2012 reported a significant increase in malate exudation and phosphorus acquisition in maize following biochar application. Additionally, microbial inoculants containing PGPR or AMF can enhance nutrient mobilization by modulating exudate composition through plant-microbe signaling interactions. Case studies in root exudate optimization. Numerous case studies illustrate the practical applications and benefits of root exudate optimization in agricultural systems. One notable example is the development of phosphorus-efficient maize varieties in sub-Saharan Africa. Conventional maize varieties often exhibit poor performance in phosphorus-deficient soils, necessitating high fertilizer inputs. Researchers at the International Institute of Tropical Agriculture identified genotypes with enhanced citrate exudation, which improved phosphate solubilization and increased grain yields by 35% without additional phosphorus inputs. In rice cultivation, the introduction of the OSPTF1 gene enhanced phosphate uptake by increasing root exudation of organic acids. Field trials in phosphorus-deficient soils showed yield improvements of 25% compared to non-transgenic lines. Similar approaches have been applied to wheat and barley to improve micronutrient uptake, particularly iron and zinc, in calcareous soils prevalent in arid regions. Ecological and environmental implications of root exudate manipulation. The manipulation of root exudates has significant ecological and environmental implications. Enhanced nutrient absorption reduces the need for synthetic fertilizers, thereby decreasing environmental pollution, greenhouse gas emissions, and eutrophication of aquatic ecosystems. For instance, phosphate exudation strategies in maize and rice have reduced fertilizer application by 30% in pilot studies conducted in India and Brazil. However, altering root exudation patterns can also impact soil microbial diversity and ecosystem stability. Excessive secretion of specific compounds might favor opportunistic microbes or disrupt symbiotic relationships. Long-term field studies by Kuzyakov and Domansky indicated that increased sugar exudation in monoculture systems reduced microbial diversity and promoted soil-borne pathogens. Therefore, optimization strategies must consider the balance between enhanced nutrient uptake and microbial community health. Future directions and challenges in root exudate optimization. The optimization of root exudate composition for improved nutrient absorption presents promising opportunities for sustainable agriculture, particularly in nutrient-deficient and degraded soils. Future research should focus on integrating advanced molecular techniques, such as metabolomics, transcriptomics, and CRISPR-based gene editing, to unravel the genetic and biochemical pathways governing exudation processes. One of the key challenges lies in the complexity and context dependence of root exudation patterns. The interactions between plants, microbes, and soil properties create dynamic feedback mechanisms that complicate predictive models. Developing robust field-based assays to monitor exudate profiles and their ecological impacts will enhance our ability to design tailored solutions for diverse agricultural settings. Additionally, interdisciplinary collaborations between agronomists, microbiologists, and soil scientists are essential to translate laboratory findings into practical agricultural practices. Public awareness and policy support for innovative bio-based nutrient management strategies will further promote the adoption of exudate-optimized crops. Conclusion: the optimization of root exudate composition represents a frontier in plant science with significant implications for nutrient absorption, soil health, and agricultural sustainability. By leveraging natural plant processes, researchers and farmers can reduce dependence on chemical fertilizers, enhance crop yields, and mitigate environmental impacts. Continued exploration of the mechanisms, factors, and strategies governing root exudation will provide novel insights and tools for future agricultural innovations, ultimately contributing to global food security and sustainable development.